Hello, and welcome to this Nature.com custom webcast titled Accelerating the Discovery of New Antimicrobial Compounds. My name is Jayshan Karpin, and I will be your moderator. Today's webcast is sponsored by LabSite. We'll begin the webcast with presentations from Dr. Riccardo Russo of the New Jersey Medical School at Rutgers and Dr. Ian Russell of LabSite.
Good day, everyone. Welcome to this presentation. I am Riccardo Russo. I'm the BSL-3 manager and senior scientist at the New Jersey Medical School of Rutgers University in Newark, New Jersey. Today we will review and discuss some of the major assays used in the first step of the discovery of new antimicrobial compound. And in particularly, we will look and discuss on strategy that can be used to speed up a screening program. In this presentation, we will first look at the major interest of our laboratory. Then we will discuss the major assays used in antimicrobial compound discovery, and especially we will discuss about automation and miniaturization of this assay using the Eco Liquid Dispenser. Finally, we will look at some managerial improvement that can be used to speed up an academic process using a Lean Six Sigma project. In my position, I wear different hats. I manage the in vitro screening core laboratory directed by Dr. Nancy Connell. I'm also the manager of the Biosafety Level 3 laboratory. It's a laboratory where we need to wear protective equipment, as you can see in the picture on the right. And this laboratory we work mainly with Mycobacterium tuberculosis. And I'm also a senior scientist, and I do a lot of the research and experiment in our laboratory. In our laboratory, the major interest of our laboratory is the discovery of new antibiotics against several high-threat bacterial pathogens. We do research to discover new antibiotics against for escape pathogens like Enterococcus faecium, Staphylococcus aureum, Klebsiella, Pseudomonas. But we also work in projects with other BSL-2 pathogens like Neisseria. We do a lot of research with tuberculosis and with other non-tuberculosis mycobacteria, and do a lot of projects with select agent that will be bacteria like anthrax, pestis, Rickettsia. We are also part of the Center for Excellence for Translational Research, and we will discuss what this center is more in the next couple of slides. And we do a lot of projects in collaboration with a major pharmaceutical company, biotech company, and several research groups from different institutions. The Center of Excellence for Translational Research at Rutgers University is funded by NIH and directed by David Perlin. This program is a collaboration between senior investigator at Rutgers University, Rockefeller University, and scientist at the Cube-based pharmaceutical, now part of Merck. The goal of this program is to identify and develop lead of a new antimicrobial compound that can be brought to the preclinical evaluation. The central program is constituted by 5 different research laboratories and several core support laboratory. There is the in vitro screening laboratory directed by Dr. Connell and me, the medical chemistry laboratory directed by Dr. Friendlich, the structural biology core laboratory directed by Dr. Liu, and in vivo animal laboratory directed by Dr. Perlin. If you are interested in getting some more information, you can go at the link at the bottom of the slide. In next slide you can see what is the normal workflow in drug discovery and development, and the central program is located at the beginning of this workflow, where there are the new candidate antimicrobial compound are identified, developed, optimized, and brought to the preclinical stage. At that point, the idea is one of the pharmaceutical company will take this candidate antimicrobial compound and start all the clinical trials. We will now review some of the major assays used in the first step of antimicrobial drug discovery, and especially we will look at a possible way for automation and miniaturization of this assay using the echo liquid handler instrument from LabSite. After me, Jan Russell from LabSite will give you a better overview of this instrument, technical particularity about this instrument. For us, it's been very useful because we are able to use much lower volumes of our compounds, and since this instrument uses acoustic waves to dispense different amount of compound, we have avoid the problem of hydrophobicity of compound, or compound sticking to tips or the walls or the wall of the wells, and we saved a lot of supplies. We use much less tips, other supplies. The minimal inhibitory concentration assay is one of the main assays done at the beginning, and is used to determine the lowest concentration of a compound that inhibits visible growth of bacteria. It can be done on agar plate or liquid broth. Most of the time 96 wall plates are used for the liquid broth assay. Compounds are tested at different concentration. Generally 1 to 2 dilution are made of the compounds. On the agar plate, you can see the activity, if a compound has any activity, by zone or inhibition around the drop or the disc or where you put the compound. In agar plates, usually you can detect by observing the formation of bacterial pellet, or can we use Alamar blue assay, that the Alamar blue is a dye that will became a path from blue to pink. Pink if you have metabolically active bacteria. It's important to consider that different bacteria can form a different shape of pellet, and sometimes we have been noticed that some compound can influence the shape of the pellet of the bacteria, like you can see in the picture on the right hand side of the slide. We develop a protocol with the echo instrument for the MSC assay. In this case, we were able to switch from 96 wall plates to 384 wall plates, and that means that one 384 wall plates correspond approximately, dependence of the experiment design, to 4 6 96 wall plate. In this case, we use as a detection method Alamar blue because the walls are too small to be able to observe bacterial pellet. And with the echo instrument, we are now able to prepare much more plates in a particular time. For example, with the echo, in 1 hour, we are able to prepare 14 384 wall plates that approximately correspond to 84 96 wall plates, and the same time that the same number of plates that we prepare in 1 hour with the echo will take us at least 8 hours to do manually. It's also important to notice that since compounds are dispensed with the echo using sonic acoustic waves, there is no problem. Compound remain stick to the tips. So there is no carryover or problem with hydrophobic compound, and that can be very important. We have noticed really only with few compounds a significant difference in the MSE when we perform the MSE assay with the echo machine or manually, and especially when we notice that if we do manually and we will change tips between each wells, at that point the result of the MSE assay will be very close or the same than done with the echo. And most of the time what we notice that the compound assayed by manually will look much more potent, so which that bigger activity against bacteria than in reality they had, and basically what we observe with the echo. And that can be as a significant, as you can see, for example, in this slide there is a table. By the echo, one compound look to have an MSE of 0.063 microgram per milliliter. When we did it by hand, changing tips, the MSE looked like it was 0.0004 microgram milliliter, so very significant difference. That could have a significant influence of for running other assay, like to test compound in animals, because a miscalculation of the MSE could interfere with the proper design of an animal study, and then with the conclusion that the compound might be not effective against that bacteria in vivo. There is also an interest in the field to use a combination of compound to treat patients. This is especially important for infection due to MDR bacteria, so multidrug resistant bacteria that are increasing in hospital setting. But also it starts to be an interest to treat other normal infection. For this reason, it's very advisable to test the possibility that new compounds have some synergic activity among themselves or with drugs that are currently used to treat particular bacteria. For example, is with tuberculosis. This assay is similar at the MSE assay, but in this case we are testing different concentration at the same time of 2 or even 3 drugs. So 2 or 3 drugs are diluted in a 96 well plate. Normally you have the first drug diluted between row and the second drug diluted between column. It is a more complicated assay to set up than the normal MSE and require more time to prepare, and is more easy to make errors. We have found that with the echo instrument we are able to prepare more plates for synergy assays, and there is less problem of error since the plates are prepared by the machine. Again, we switch from 96 to 384 well plates, and always depend from the experiment design. Usually we test 9 dilution of both drugs. One 384 well plate correspond approximately a 6 96 well plates. Again, we use Alamar Blue as detection method. And with the echo we are able to prepare in 1 hour 8 to 10 384 well plates that will correspond to 50 to 60 96 well plates. It will take 24 hours to prepare the same amount of plates manually. So it's been a big advantage, the echo, for us, because we are now able to run regularly synergy assay, meanwhile before we were just running in some occasion. Another important assay to run is the MBC assay, that stands for Minimal Bactericidal Concentration Assay. This assay is like the MSE, but at the end of the assay we go to determine the actual number of bacteria in each well. This is an important assay because it can help us to determine if a compound is bactericidal or bacteriostatic. With the result of the MSE assay, we determine a concentration of bacteria of a compound that inhibit any visible growth bacteria, but we don't know if the number of bacteria from the beginning of the assay, from the inoculum, has gone down, remain the same amount of bacteria, or slightly increased. The only way to determine if the bacteria from the inoculum have been killed, or how much bacteria have been killed, or they just didn't divide, is doing an MBC assay. So at the end of the experiment, or MSC experiment, plate each concentration of compound and determine the amount of bacteria present. So clearly serial dilution will be done from each well, or sometimes you can pull up the replicates of a treatment, and then there are all the serial dilution are plated on agar plate. As you can imagine, it's very time consuming assay and very work intensive. Luckily we are able to develop an application of the MBC, or better the CFU assay, that colony forming unit, to determine the amount of bacteria in the liquid. We are able to develop an application with the echo. When we want to determine the concentration of bacteria in each well of our 96 well plate that we use for the MSC MBC assay, what we normally do, prepare several 1 to 10 dilution of each well, and normally we prepare even 8 dilution of each well, and then we need to plate each dilution on agar plates. With the echo, instead, what we do, we just prepare 3 or 4 dilution of each well. We put those dilution, we load for example in a 384 well plates, and the echo will automatically dispense different amounts from each of the 4 dilution on a uni well plate. So it's like a 96 well plate that has only one big well, that where we pour agar. So in this way we are able to plate 48 samples in 8 minutes. Manually will take almost 4 hours to make dilution of those 48 samples and to plate them. So we significantly reduce the time and the material required to perform these CFU assays. Once we determine the MSC values of new compounds, it's important to determine if the plasma protein can bind the compound, and so they can significantly influence the biological activity of new drugs. For this reason, it is advisable to run the serum binding assay. This assay is similar to the MSC assay, but we add between 10 to 40% of serum to the media. It's important to consider which serum to use. Normally this assay is run with bovine sera, but if you are going to test our compounds in animal models that most likely are not bovine, so will be mice, and also this compound will be used in human, it's important to run the serum binding assay with mice and human serum. In our experiment, we have noticed in several cases that there was a significant difference in binding capacity among bovine, mouse, rabbit, and human serum. So if we run this assay only with bovine serum, for example, and we are going to test the compound in mice and then in human, we could have different results between the in vitro and in vivo experiment. For example, for one compound we found no great binding activity of bovine sera, but when we test the compound in mice, we noticed that the compound was not as much effective in vivo models. And when we went back and rerun the serum binding assay with mice sera, we noticed that the reason was that the compound was significantly bound by mouse sera, but not by bovine sera. Next important assay to run is the mammalian cytotoxicity assay. It's important to determine that if the compound can be toxic to different mammalian cell lines. Often we observe very potent compounds that have a very strong antibacterial activity, but unfortunately they were also very toxic to mammalian cells. So it's important to determine the concentration which the compound is toxic to mammalian cells, and ideally we want a big difference between the toxicity level and the level at which the compound is active against bacteria. We are working now on developing an application for the liquid handler to run cytotoxicity assay quickly. If you are trying to develop or discover a new compound that have an activity against bacteria that can survive inside mammalian cells, for example, think to Mycobacteria tuberculosis, Salmonella, Francisella, Rickettsia, that are all bacteria that are able to survive inside the macrophage or other cell line. It's very important to determine if your compound will be active against these bacteria once they are inside a mammalian cells. So if the compound is able to enter the mammalian cells. You can use as detection in this case, you will, in this experiment design, you will infect mammalian cells with your bacteria, and there are different ways how you can determine how many bacteria are at the beginning of the experiment and after the treatment with your compound. The classical way is to lyse the mammalian cell at the end and plate for CFU, or you can run a PCR, or you can label the bacteria with fluorescent or luminescence proteins, so you can read the fluorescent and luminescence. We are working on developing application for this type of assay with the echo handler. In this slide, we present the result of an assay we developed transforming Rickettsia canadensis with green fluorescent protein, and we detect the activity of different compound reading fluorescence. In this case, clearly is much cheaper and faster to detect the presence, the concentration of bacteria in the cells using fluorescence compared to the classical PCR techniques. So one of our grad student, Alex Lemense, ran this assay, developed this assay, did a cost analysis, and we saw that there was like a decrease almost like a 96% in the cost of running this fluorescent assay compared to a PCR detection method. I want to give you just a few final considerations regarding this assay. It's important to test a new candidate compound against pathogenic bacteria, so Pseudomonas, MRSA, any of the escapes or tuberculosis or select agent. Happened to me sometime to review articles where compounds were only tested against laboratory strain of E. coli or some non-pathogenic or very low virulent bacteria. So it's important instead to test this compound, the new candidate compound, against pathogenic bacteria. Give more importance to your paper, and it's clearly more important to find new candidate compound that can be used against high-threat bacteria. It's also a good experiment designed to test not only against one bacteria but a panel or strain of one species of bacteria. In this case, it's good to use different laboratory strain, and especially test the compound against several clinical strains of this bacteria. It's also very advisable to test multidrug resistant strain of this bacteria. Clearly, as we discussed before, it's very important to determine if the compound is bacteriostatic or bactericidal. So, if your compound is just inhibit the growth of the bacteria, or if it's really killing the bacteria. In that case, what percentage of bacteria are killed? And it also is important to run synergy assay. We now finished the first part of this presentation where we review different assays, and we are now going to the next part where we will look at some management adjustment that can be done to increase the efficiency of an academic screening program. And we use a Lean Six Sigma project to determine how we could improve the accuracy of our assay, decrease errors, decrease the time from when we receive compound to when we submit the compound to the different groups. So remember that in our laboratory we are a core lab, but we also do research. We are an academic institution and not an industry, so there are several constraints clearly. And we receive hundreds of chemical compounds every month from several research groups, and we run a lot of different assays and with a lot of different bacteria. Many people are involved in this process. So in this project we want to try to put some goals that were to try to decrease the time to test the compound and the mistake errors of that compound that needed to be tested again. To get a better understanding of the process of testing compound in our laboratory, we start to design a flow map at the beginning of this project. So all the steps involved from when we receive the compound to running the assays and submitting the results. We start after that to run several other tools that are used in Lean Six Sigma project, like Pareto charts, fishbone diagram, and we came out of design a new final flow map of this process. In reality, we went to between two other iterations of this flow map, and we tested every time how it was working, and then we tried to improve. As you can see, the flow map became more complex, but reality helped us to streamline and fastening this process of testing compound and make sure to check for mistakes and be able to resolve any mistakes or any problem right away. And this allowed us to decrease the time from when we receive compound to when we submit the result of the assay. It depended again from the bacteria, but for example, for some bacteria like tuberculosis, we pass from running assay and from a time of receiving compound to submit a result of 2 months to 2 weeks. For some of the escapes pathogen, for example, we pass from a time of 14-18 days to 4-5 days, so a significant decrease in time. One thing that we created and implemented the right way is the submission forms. Before, we will have people from pharmaceutical company or in other research group just sending us an email saying that they are going to send tot number of compound that they want to be tested with this with some bacteria or some assay, and sometimes we were getting confusing after some time to determine what assay should have been run. If it was run, if we run all the assay, we tested all the bacteria they requested at the beginning. So for this reason, we create a submission form where we specify who is sending the compound, what compound are sent, the concentration they want to be tested to, if they are, for example, heat sensitive or light sensitive, if I have a problem of solubility. And in this form they can check exactly which assay they want to run against the specific bacteria they are interested in. And we have in the form 2 a list of assays they can choose through. So you can see in the first the blue arrow, we have where we indicate the field where they will write who is sending the compound and what assay they can choose from. Then in the green arrow is the place where they will write the list of compounds they are submitting with all the information regarding each compound. And then on the next section, for example, where indicated by the red arrow, they will indicate which bacteria they want to use for their assay. We then create a database that we share with all the people involved in testing compound in our laboratory, and we create a specific folder for each group from where we receive compound, and we share with somebody in that group the same folder. And in this folder we create what we call a master sheet, so a big Excel file where we report all the compounds sent, the date when the compound were sent, by who was submitted. Let me see now in green light, in green arrow, all the assay that were requested for each compound. And like you can see now by the red arrow, we also write in this master sheet the result we obtain for each assay, and in the green in the blue arrow when we submit back the result. In this way we have an idea if there is any assay that still need to be run, how long it took to run an assay. And since sometimes we get groups that will submit a compound that was submitted maybe a week, a month, a year before, we can also quickly look back and determine what were the results of the assay run before, and determine if there could be some problems. In this Lean Six Sigma project, we have done also other things, but at the end of the project, we really were able to significantly decrease the time from when we received compound to when we submitted the result, increasing enormously the accuracy of our result. A major we improved the communication among all the stakeholders, so all the members of the laboratory, but also the people that will submit the compounds, other people involved in this project, and increase significantly the capacity of running assays. So we are able now to run at least 50 times more compounds than before. We are able to easily perform much more assays, and we have more people involved in running assay, and we are getting compound from more groups. Like for example, at this moment we have 7-8 groups that send us compound, submit us compound to be tested, and at the beginning we had only 2 groups. I want to thank our PI and boss, Dr. Nancy Connell, and all the people involved in testing the compound in our laboratory. We have Margherita Tachman, Harry Singleton, Thomas Keane, Jim Ochi, Alex Lemense, Melissa Talawe, Michael Zuen, and Chi Tang. And I also want to report all the funding sources for our project, NIH, DARPA, GSK, Jacobus Pharmaceutical, and ISMED. Okay, I'll be wait now for the next section when we'll have a question and answer.
Thanks, Riccardo. And now over to Ian.
Hi, good morning, everyone. Over approximately the next 10 minutes, I'm going to give you a brief introduction into LabSite as a company and go into a little bit more detail on the Echo Liquid Handler product line and the underlying technology that drives it. LabSite was founded in 2000 and became the first company in the biotechnology field with the launch of the first Echo Liquid Handler in 2004 to transfer liquids using sound energy. In 2011, LabSite launched a proprietary technology which we call dynamic fluid analysis. And this enables echo liquid handlers to automatically adjust to different fluid types. Perhaps more importantly, however, this has enabled the company to expand beyond the traditional drug discovery workflows. And working closely with customers, particularly in the genomics space, we launched the Echo 525 in 2013, which could build larger volumes faster. And this has become the preferred system within the genomics applications where larger reaction volumes are required. To support the completely automated Echo handler workflows, I also wanted to point out that we offer complementary automation systems that are easily tailored to support a wide variety of workflows out there. The Echo Liquid Handler today now constitutes a family of instruments. On the left-hand side here, we have the Echo 525. It is the instrument with the 25 nanoliter droplet. The two instruments on the right-hand side, in fact, have the 2.5 nanoliter increments and share all the same capabilities, with the exception that the Echo 65XT, which was launched earlier this year in February, also accommodates a brand new acoustic sample tube, which was launched at the same time. Below this, I have the various consumables that are available to support the Echo workflows, all the way from a reservoir up through a 1536 well format. Those come in both standard and low dead volume flavors, as well as with various coatings to support different workflows. And what is not shown on this particular slide, but I'm not going to go into any detail, is a wide variety of software that is designed specifically to support the workflows that our customers are interested in. At the heart of the Echo Liquid Handler is an acoustic transducer, and that's depicted at the lower right-hand side of the slide. The acoustic transducer is responsible for generating the acoustic energy. It also serves the function of focusing that energy at the surface of the fluid being transferred. We have a coupling fluid that enables the acoustic energy to travel from the transducer into the particular fluid that we are transferring. And in doing so, we can generate droplets of either 2.5 nanoliters or 25 nanoliters and eject those from a source well up into an inverted destination well. For volumes that are greater than either the 2.5 or the 25 nanoliter, we're essentially just ejecting multiple droplets in succession. And those droplets can be ejected into either a dry well or a well that already contains some fluid in it. That ejection process is extremely rapid, so we can eject those droplets at rates anywhere between 200 to 500 droplets per second. We can also position that acoustic transducer under any of the wells of the source plate and position any destination well above the transducer. And so that enables us to do any well to any well transfer. As I mentioned earlier in my presentation, dynamic fluid analysis is proprietary to LabSite and has the unique benefit to the customer that it automatically adapts to fluid properties within the well and adjusts in real time as it's doing the transfer. It borrows from medical ultrasound as well as sonar. And if we view the video here, what you can see is that there is a low energy ping that's being emitted by the transducer. It's reflecting off three surfaces. First is the bottom of the plate, the second is the bottom of the well, and the third one is the surface of the fluid. And utilizing those reflections, we can infer two aspects to the fluid. The first one is the fill height of the fluid, and so that enables us to appropriately focus the acoustic energy on that meniscus. The second one is to understand the acoustic impedance of the fluid which is contained within the well. And that acoustic impedance basically describes how sound will travel through the liquid and enables the instrument to adjust to changing fluid properties as it moves from well to well. So what that means is that the instrument can accommodate a very wide range of fluid types, even within the same plate. And that could span anything from cell lysates to various reagents that are used in genomics or other applications. Those reagents could be at different concentrations. They could contain glycerol, surfactants. They could contain biomolecules such as DNA, RNA, or protein. So the advantages of the Echo liquid handler really fall into four major buckets here. The first one is maximizing that lab budget, and that's primarily through that miniaturization, but also the elimination of consumables such as tips. The second one is data quality, the ability of the instrument to very highly accurately transfer a predetermined volume and to do that very precisely. Is going to result in very consistent assay results time and time again. The speed at which the system is capable of transferring liquids, the ability to do any well to any well transfer, of course, translates into a very rapid assay design process. And then lastly, the need to automate complex workflows and the increased precision and accuracy that comes through that automation, as well as the ability to free up time for perhaps other higher value activities within the lab. I just want to give you a flavor for the extent of miniaturization that is possible with the Echo liquid handler. The examples that I'm showing here are from the genomics field, but I think they give you a perspective on what is feasible anywhere up to 100-fold. And of course, this is going to be direct savings in terms of reagents, but also if you are utilizing very precious samples, then that sample is just going to go that much farther for you. Okay, so just to quickly summarize, I mean, we've talked about the different flavors of Echo liquid handler that are available. All of those are ready to go out of the box and do not require expertise in robotics or liquid handling to make use of those. They all readily integrate into automation systems, both provided by LabSite or third parties. And I think what's important to take away with you is the transfer that you get with an Echo liquid handler is contact-free. And so that directly translates to higher data quality. There's no tips that are required for that transfer. It's highly flexible, so we have the ability to transfer from any well in the source plate to any well in the destination plate. And that enables rapid cherry picking types of applications to be performed. And it also allows steps potentially to be eliminated from your workflows. So because of the very low volumes that the Echo liquid handler is capable of working with, in many instances, pre-dilutions become unnecessary. And lastly, hopefully I convinced you that the Echo liquid handler has the potential to really help with assay miniaturization within the workflows that you're using. So with that, I'd like to thank you for your attention and turn this back over to the moderator.
Thank you for your presentation, Ian. It is now time for the Q&A with Ian and Riccardo. So our first question, and this one is for Riccardo, and it asks, due to the way tuberculosis growth in aggregates, is it possible to use the Echo liquid handler to inoculate plates with MTB?
Yes, the problem with tuberculosis and mostly a lot of the mycobacteria that they aggregate when they grow, so they are in growing clusters. So it's more complicated to calculate the exact accurate number of cells in a culture. we have not used the echo for dispense tuberculosis because we should have moved the machine inside the BSL-3 facility. And currently we are planning to do that in the near future, and we are trying to also get approval from our institutional biosafety committee. And as you can imagine, we will need to make sure that prove that we don't aerosolize any bacteria. In any case, I tested the echo with Mycobacterium magmatis, so bacteria BSL1, BSL2 level. And what I did is the sample that I was going to plate for colony count, I had 10 times more twin. The twin help a bit to break the cluster of bacteria. Say that, I will also say that if you want really accurate quantification of the bacteria, so you want to be, for example, know if they are 2 or 5 or 8, 10 to the 5, for example, I will use the manual protocols. Instead, with Mycobacteria, it's very useful when you do want to do a screening and have an idea, okay, the amount of bacteria in your sample is 10 to the 5 or 10 to the 4 or 10 to the 7. So like a sort of a screening qualitative results. I hope I answer your question. But with TB, we are going to soon try to do that with TB. Tuberculosis.
And our next question asks, is there any way to check intercellular MDR or MDR efflux pump mechanisms? And can you give your suggestions and comments on that?
I haven't not tried. I imagine what you could do is having a culture. So, for example, we have just used the echo for cytotoxicity assays. So we have the plate with a layer of Vero mammalian cells with 50 microliter of media, and we are able to add to those wells compounds that you want to test for cytotoxicity. So what maybe you can have is having the bacteria and then add your compound, your chemical you're trying to determine if they have any effect on a flux pump. But usually, from what I know, it's a more complicated type of experiment design than just add a compound. So I'm sorry, I cannot. I don't know really. Maybe we can discuss later with this person by email.
Could you elaborate on how you prepare your source plate for your MBC dilution plate on the echo?
Yes. So what I do is we are writing really an article to describe in all the detail this protocol and some of the other protocols that we develop. But probably what I do is I prepare 2 or 3 1 to 10 dilutions of the original culture. So I have the original culture, I do 1 to 10 in brown culture, 1 to 10 dilution, then I do a second 1 to 10 dilution and another 1 to 10 dilution, and then use the echo to dispense different volume from each of the 4 cultures, so the original one and then the 1 to 10, 1 to 100, 1 to 1000 dilution, and using the echo I dispense different amount of those culture on a big agar plate. So usually we use what is called a uni-well plate. It's like a 96 well plate, but it has only one big well, and so we fill it up with agar, and then we shoot the transfer bacteria from the cultures to that plate. And we put the original culture and all the dilution in, we load in one of the plates from the echo that is a 384 source plate.
And our next question, I think this one's for you, Ian. And it asks, how do the inverted wells retain the liquid?
Yeah, I can certainly address that question. It's a very common question. It's a unique setup within the echo. The basic premise here is that the surface tension of the liquid within the well is sufficient to retain the fluid within the well. When customers first start with the echo, that is an area of concern on their part. And we commonly just recommend that they do a test run with a particular plate and fluid type that they're using, turn it over, tap it on the bench above a Kim wipe or something like that, and just confirm that nothing falls out at the volumes that they're working with. But I will say very quickly they get comfortable with the ability of the plates to retain that. What will obviously affect the surface tension are some of the common additives. So, for example, surfactant will lower that surface tension. And so one just needs to be a little bit careful and confirm that the volumes are not going to result in any liquid dripping out. But commonly, the volumes that we're working with do not cause any issues there.
Asks how many compounds are assayed per year, and have any of the compounds moved into the development phase of the CETR workflow?
So in our laboratory, we receive a compound from many different institutions and pharmaceutical biotech company. We do not only do this type of assay. We have a lot of other type of experiments we run, but usually we run around 10,000 compounds. We receive 10,000 compounds that are tested against several different bacteria. So we run several different assays, and we have some lead candidates in our central program that are sent toward the preclinical, are considered now for preclinical studies. But as you say, it's everything. I cannot say anymore because it's everything in research going on, so nothing has been published.
What types of liquids can the echo transfer, and does it require calibrating for each liquid type?
Yeah, I think that's a very good question. So LabSite takes a lot of effort in evaluating different liquids and defining calibrations for them. So when a customer takes receipt of an echo liquid handler, it is pre-calibrated. We have specific calibrations for certain fluid classes. So just at a broad level, we have calibrations for DMSO. And then we have, within the aqueous realm, a couple of specific calibrations dependent on the makeup of the fluid that you have there. And it's typically broken out in terms of standard buffer, and then whether it contains glycerol or whether it contains surfactant. We do also have a broad calibration. So if you have a fluid where it doesn't fit into one of those particular classes, or you are unsure of what the contents are, we have a broad spectrum calibration that spends more time interrogating the fluid in there and making adjustments on the fly. The downside to that particular calibration is a little bit slower, but it is able to accommodate a wider range of fluids. It would also be useful if you have a plate that has fluids from a variety of those different fluid classes that I just described, as it is able to adjust across all of the different, whether it contains glycerol, surfactant, DMSO, etc.
Is it possible to send a compound for testing? And if so, how can you do it?
Yes, they can send us compound for testing. It's easy. They just needed to contact me.